Hi, my name is Pamela Coons, Associate Professor of Medicine in the Division of Oncology at Yale School of Medicine and Yale Cancer Center. I'm excited to announce ASCO's new open access journal, JCO Oncology Advances. As the inaugural editor-in-chief, I hope to support JCO Oncology Advances to become the premier platform to bridge the gap between accessible scientific research and clinical care. Stay tuned for more information, including new article types, at ascopubs.org/JCO Oncology Advances. We look forward to seeing your submissions in spring of 2024.
This JCO podcast provides observations and commentary on the JCO article 3 years follow-up of an Alectinib phase 1/2 study in ALK-Positive non-small cell lung cancer, AF-001JP, by Tamura et al. My name is Ben Solomon, and I am a medical oncologist at the Peter MacCallum Cancer Centre in Melbourne, Australia. My oncologic specialty is thoracic oncology. The presence of ALK gene rearrangements in about 3 to 5% of non-small cell lung cancers molecularly defines a subset of tumors characterized by sensitivity to ALK tyrosine kinase inhibitors. Phase 3 studies have shown that crizotinib, the first-in-class ALK inhibitor, results in higher response rates and progression-free survival than chemotherapy, establishing crizotinib as standard of care for this patient population. However, after a median of approximately 11 months, patients treated with first-line crizotinib develop disease progression, with the brain being a frequent site of failure. Alectinib is a highly potent and selective second-generation ALK tyrosine kinase inhibitor. It is not only a more potent inhibitor of wild-type ALK than crizotinib, but it has activity against many mutations in the ALK tyrosine kinase domain that confer resistance to crizotinib, including L1196M, the so-called gatekeeper mutation. Alectinib also shows excellent CNS penetration and intracranial activity in preclinical models. The AF-001JP study was a phase 1/2 study of Alectinib conducted in Japanese patients with ALK-rearranged non-small cell lung cancer who had not received prior ALK TKI therapy. A recommended phase 2 dose of 300 milligrams BID was determined in the phase 1 component of the study. In the phase 2 component of the study, a remarkable objective response rate of 93.5% was reported in 46 patients treated at 300 milligrams BID, which included 2 complete responses and 41 partial responses. These data led to the approval of Alectinib in Japan in 2014. The article by Tamura and colleagues that accompanies this podcast provides an update for efficacy and safety more than 3 years after enrollment of the last patient on study, and confirms the long-term durability of responses to Alectinib in this population. At the time of this report, 25 of 46 patients enrolled on Part 2 of the study treated at 300 milligrams BID were still receiving treatment, progression having been noted in only 18 of 46 patients. The median progression-free survival has not yet been reached, and 3-year progression-free survival for all Phase 2 patients was 62%. The authors attempted to correlate tumor shrinkage with progression-free survival, but did not identify any correlation between depth of response and progression-free survival. The median overall survival was not reached at the time of report, and 3-year overall survival was 78%. No new toxicity signals were identified with the additional follow-up. Even with long-term daily administration, most treatment-related adverse events from Alectinib were mild to moderate. Of note, the 300 milligrams BID dose used in this Japanese study is different to the Alectinib dose used in the rest of the world. In AF-001JP, 300 milligrams BID was the highest planned dose, and although no dose-limiting toxicities were seen, no further dose escalation occurred due to the concentrations of an excipient called sodium lauryl sulfate within tablets. In a subsequent phase 1/2 study conducted in the United States, 600 milligrams BID was found to be the recommended phase 2 dose, and this dose has been used in subsequent global phase 2 and phase 3 studies. Plasma levels achieved with the 300 milligram BID dose in Japanese patients appeared to be similar to levels reached in Western patients using 600 milligrams BID. Another important difference between the Japanese phase 1 and the US phase 1 was that the Japanese study was conducted in patients that had not received prior ALK tyrosine kinase inhibitors, while the US phase 1 study enrolled patients whose tumors had progressed on prior crizotinib. The objective response in the crizotinib refractory population enrolled in the US phase 1/2 study was 55%. This was in comparison to the 93.5% seen in the AF-001JP study. Two further phase 2 studies, one conducted in North America and the other globally, reported response rates of 52.2% and 50% in patients who had received prior crizotinib, leading to US FDA approval in 2014 of Alectinib for patients who had progressed on crizotinib. The CNS activity of Alectinib is of considerable interest given the frequency of brain metastases in patients with ALK-rearranged non-small cell lung cancer, and the observation that the CNS is a frequent site of progression on crizotinib. Brain metastases were present in about 1/3 of patients enrolled in this study. 6 of the 14 patients with brain metastases at baseline remain on study without evidence of CNS or systemic progression. The median progression-free survival in all patients with brain metastases was 38 months. The CNS activity has been further evaluated using pooled data from a North American and global phase 2 studies and previously reported by Gadgil and colleagues in JCO. Results from 136 patients with baseline CNS disease, comprising 60% of the overall pool population, showed frequent and durable responses. In patients with measurable CNS disease, 32 of 50 patients had intracranial responses, with 11 patients, or 22% of the population, developing complete responses indicative of significant CNS activity of Alectinib. In summary, long-term follow-up of AF-001JP indicates that the responses to Alectinib are not only frequent, with an objective response rate of 93.5% in crizotinib-naive ALK-positive patients, but are durable and associated with progression-free survival in excess of 3 years. The activity of Alectinib, and indeed other second-generation ALK TKIs such as ceritinib, brigatinib, and ensatinib, raise questions about how these agents should be optimally sequenced, and in particular about which agent should be used in the first-line setting for newly diagnosed ALK-positive patients. This question is addressed by 2 phase 3 studies that compare first-line Alectinib to crizotinib, the existing standard of care. The first of these, a Japanese study called J-ALEX, was strongly positive in favor of Alectinib. The median progression-free survival was significantly improved with Alectinib with a hazard ratio of 0.34, and the objective response rate was 91.6% for Alectinib compared with 78.9% for crizotinib. If a second global study, the ALEX study, confirms these results, Alectinib will be established as a new global standard for first-line treatment of ALK-rearranged non-small cell lung cancer. This concludes this JCO podcast. Thank you for listening.
For more original research, editorials, and review articles, please visit us online at jco.org. This production is copyrighted to the American Society of Clinical Oncology. Thank you for listening.